welcome to the Deep Dive. Today we're plunging into a truly transformative area, digital health technologies or DHTs, and their, well, explosive impact on clinical trials. Imagine unlocking continuous real-world data from the devices you carry every day. Your smartphone, your wearable. That's the revolution we're witnessing.
Yeah, and it's far more than simple convenience. We're not just talking about, getting a step count.
Right.
This is about capturing incredibly rich, high-frequency information that could fundamentally change how we understand health, how diseases progress, and critically, how treatments actually work in people's daily lives, not just in a clinic visit.
I understand. And that clarity about our mission is crucial for this deep dive. Our goal today is to really unpack the critical challenges and maybe more importantly, the ingenious solutions the scientists and regulators are grappling with, all to leverage these DHTs for robust, regulator-approved clinical trial endpoints. We're going to cover everything from managing those inevitably messy data streams
Oh yeah, messy is the word.
to a landmark case.
Yeah.
The very first digital endpoint to actually get the green light from regulators.
Exactly. Think of this as a journey into how science is adapting to the digital age. It's about ensuring that the incredible amount of data collected from your personal devices can genuinely accelerate medical progress and bring new, effective treatments to patients faster than ever before, really bridging that gap between everyday tech and groundbreaking medical discovery.
Okay, all right, let's unpack this by starting with, well, an inevitable hurdle. While DHTs promise this amazing continuous stream of data, real-world data collection, it always suffers from missingness, doesn't it?
Always. It's unavoidable.
And it's not just a blank spot. It introduces significant statistical bias. So we're talking about missing data and this bias issue. What's the most challenging type of missingness you encounter, and why is it so problematic for study results?
That's a crucial question. Look, when we talk about missingness, it can appear in a few ways. You've got epoch-level gaps that's missing seconds to maybe hours of data.
Yeah.
Then there are daily summary gaps where you might miss entire days of, say, step counts. And of course, participant-level attrition, which covers dropouts, or when someone just stops wearing their device altogether.
Right, they just fall off the map.
Exactly. But the most insidious type, the one that really causes the most trouble statistically, is often missing not at random, or MNAR.
MNAR. Insidious is a strong word for data. Why? Why is it particularly sneaky?
It's sneaky because it happens when, for instance, participants take off their devices because they're feeling unwell, maybe too fatigued.
Ah, I see.
Or even sometimes because a treatment is making them feel much better and they just forget to wear it or don't feel they need it anymore.
Right.
So if that data is missing because of the patient's health status, it can seriously bias the results. It might make an endpoint look better or worse than it actually is in reality.
Okay, that makes sense.
And this is why standard methods, the old ways like just carrying forward the last observation, LOCF, or simply deleting incomplete data, they're often insufficient, even problematic.
Because they don't account for why it's missing.
Precisely. They ignore the underlying reason, which might be directly linked to the outcome you're trying to measure.
So if simply deleting data or LOCF isn't good enough, what clever statistical strategies have researchers developed to address this? How do you fill those gaps more intelligently?
Well, we have several approaches. For those smaller, epoch-level gaps, we can often use within-patient imputation.
Right.
This basically involves replacing missing bits by looking for patterns from the same participant, maybe using the median or mean value from similar times of day on other days for that specific person.
So you're leveraging their own data history.
Exactly. It keeps it personalized.
Makes sense. Or I read about functional principal component analysis, FPCA. That sounds complex.
It is a bit more sophisticated, yeah. Imagine you've got movement data, like tracking gait, and parts are missing. FPCA is like a smart curve-fitting tool. It treats each person's data over time as a sort of smooth function. It understands the natural flow, say, of walking patterns, so it can intelligently predict and fill in those missing segments to reconstruct a continuous, natural-looking curve. It's quite powerful for things like motion data. And then for summary-level endpoints, like, say, total daily step counts, where maybe a whole day is missing, multiple imputation, or MI, is a really robust technique.
How does that work?
It leverages other patient data covariates, maybe their step counts from previous days, across the whole data set.
Okay.
It doesn't just create one best guess for the missing value, it creates several plausible, complete data sets.
Right.
Then you analyze all those data sets and pull the results. This approach better reflects the inherent uncertainty around that missing data point.
Got it. And what about the really complex patterns, like long-term trends? I hear about deep learning approaches.
Yeah, things like time series neural networks, maybe LSTM autoencoders, these are like super smart pattern detectors.
Hmm.
If a person typically takes, say, 3 fast steps, then slows down, these models can learn that sequence. So if some steps are missing in the middle, they can infer what likely happened based on the patterns before and after.
Very sophisticated.
Very. But there's a crucial caveat with deep learning. They need a lot of data and long training times, and you have to be really careful about fairness, making sure the model works well for all patient groups and doesn't inadvertently introduce bias based on how well represented different groups are in the training data.
Right. Fairness is key. Now, beyond fixing the data after it's missing, what about preventing it in the first place? Proactive measures.
Absolutely critical. Proactive mitigation in the protocol design is equally key. This really starts right at the beginning with robust participant training and onboarding.
Like a practice run.
Exactly. Using run-in sessions helps ensure people are comfortable with the devices and know what's expected before the official data collection starts. It boosts compliance right from the get-go.
And once the trial is actually underway, interactive compliance tools must be vital. Real-time feedback seems way better than finding out later.
Spot on. We're talking apps that send notifications if a device hasn't been worn for a while.
Okay.
Or real-time dashboards that study staff can monitor. If they see someone's adherence dipping, they can reach out promptly, see if there's a problem. Maybe the device isn't charged or they're having trouble.
Catching problems early.
Exactly. And another really important aspect here is device equity.
Meaning?
Meaning providing devices, maybe even basic smartphones or data plans, for participants who might lack the compatible technology. This helps reduce inherent bias in who can even participate in the trial.
Yeah, avoiding that digital divide.
Precisely. It ensures a broader, more representative study population, which makes the results more generalizable.
And then when it comes to the actual analysis, you mentioned pre-specifying wear time thresholds.
Yes, that's crucial. You need to define upfront what counts as enough data. For example, requiring maybe at least 10 hours of wear per day on at least 5 out of 7 days a week, that becomes your minimum standard.
Okay.
And then it's absolutely imperative to conduct sensitivity analyses. You have to test how robust your findings are under different assumptions about why the data might be missing.
Like assuming it was missing completely at random versus missing at random versus that tricky M&AR scenario.
Exactly. You run the analysis under each assumption and see if your main conclusions still hold up. It's about stress testing your results.
And I imagine the regulators are very keen on seeing this.
Oh, absolutely. That scrutiny isn't just good scientific practice. It's something regulators really demand. Bodies like the FDA and the European Medicines Agency, the EMA, they require these pre-specified missing data handling plans.
Right, the analysis plan.
Right, in the statistical analysis plan. Sponsors have to clearly demonstrate how their imputation methods preserve the integrity of the endpoint without introducing bias. It's basically a non-negotiable part of getting these digital endpoints accepted for decision-making.
That's a really robust approach to ensuring data integrity. But the journey of digital health in trials isn't just about handling messy data. It's also about where that data comes from. Let's pivot to a major trend. Bring your own device. BYOD.
Oh, yes, BYOD.
It seems like such a natural fit, doesn't it? You get to use your own phone, your own watch. What are the big opportunities here?
The opportunities are pretty significant, actually. First off, BYOD can increase participant compliance. People are just more comfortable and familiar with their own tech.
Makes sense.
It can also potentially reduce the Hawthorne effect, that phenomenon where people act differently just because they know they're being observed or using a special study device.
Right, they act more naturally.
Exactly. And, importantly, it can lower study costs. You're not buying devices for everyone. Plus, it allows for data collection in truly diverse real-world settings that better reflect how people actually live their lives.
Okay, BYOD sounds fantastic on paper, but I imagine it brings a whole new set of headaches. What would you say is the single biggest challenge when you're trying to integrate this huge variety of personal devices into a rigorous clinical trial?
You've really hit on it. The heterogeneous tech landscape. It is potentially a data scientist's nightmare.
Huh.
You're dealing with data variability coming from countless different devices, different brands, different sensors, different operating systems, different software versions.
Right. It's not like everyone has the same iPhone and Apple Watch.
Not at all. You've got everything from a brand new flagship phone to a 3-year-old budget Android, all supposedly feeding comparable data into your study. It's like trying to orchestrate a symphony with, a kazoo, a violin, and a tuba all playing the same note.
Okay, so that variability is huge. What else?
Another major challenge is digital literacy and access bias. There's a very real risk of underrepresenting certain groups, maybe older adults, people in rural areas, lower socioeconomic groups if participants lack compatible devices, or the digital skills needed to use the apps or troubleshoot issues.
The digital divide again.
Exactly. And we should also acknowledge that while regulatory guidance on BYOD is definitely emerging, it's not yet fully mature. The rulebook is still being written to some extent.
So given that complexity, how do these BYOD trials actually deploy the technology? Are there different models?
Yeah, there are basically 3 main configurations you see. First, the simplest is just a smartphone-based app, where the app itself collects data using the phone's built-in sensors like accelerometers for movement, maybe GPS.
App only.
Right. Second, you have the hub model. Here, the smartphone acts as an intermediary, like a data hub, connecting to external sensors, maybe a separate wearable, a smart scale, a blood pressure cuff via Bluetooth usually.
Okay, phone plus external sensor.
Yep. And finally, you have the standalone DHT. This would be a device like maybe an advanced wearable with its own cellular connection, like an eSIM, that can transmit data directly without needing a smartphone nearby.
Got it. So how do you mitigate those bias issues you mentioned, the access problems?
Well, a key strategy is to offer provisioned devices, study-provided devices, or maybe internet support, like a mobile hotspot, for participants who lack access.
So a hybrid approach sometimes?
Often, yes. You might aim for BYOD, but have provisioned devices as a backup or for those who need them. It's also crucial to diversify recruitment strategies to actively reach those potentially underserved populations.
That makes sense. And on the data handling side for BYOD?
Oh, capturing metadata is absolutely essential. You need to know the exact device model, the operating system version, the app version, maybe even firmware versions of sensors.
To understand the context of the data.
Exactly. It helps you analyze potential variability later. And those real-time compliance dashboards we talked about, still critical here, maybe even more so with BYOD, plus having alert systems for drop-offs. And of course, a huge piece is addressing privacy and consent, especially for passively collected data from someone's personal device. You need crystal clear consent processes. Trust and transparency are absolutely key.
What's the regulatory stance on BYOD been like? Are they encouraging it or cautious?
It's interesting. The FDA, for instance, actually encourages sponsors to consider using participants' own technologies. They've even stated they haven't found consistent differences in compliance between BYOD and provisioned device studies.
Oh, interesting.
Yeah, but they still strongly recommend maintaining provisioned device options precisely to avoid that potential bias and ensure everyone who's eligible can participate.
So it's about finding that balance.
Exactly. Leveraging the advantages of BYOD, the convenience, the real-world context, while carefully safeguarding data integrity and participant representation. It's about finding that sweet spot between convenience and scientific rigor.
Okay, so we've dug into the practicalities of data, missingness, devices, but before we can even really start measuring things effectively, we face a surprisingly fundamental challenge. It's less about hardware or software, more about just shared understanding. The language barrier.
Ah, yes. Terminology.
Why does the very terminology we use or misuse create such a hurdle in digital endpoint development?
It's a massive challenge, honestly, because the confusion caused by inconsistent terminology, you hear terms thrown around like digital measure, digital biomarker, digital endpoint, ECOA,
Right.
and often people use them interchangeably or have slightly different understandings. It means everyone is speaking a slightly different dialect, if you will, and that leads to real problems down the line.
What kind of problem? What are the consequences of that ambiguity? It seems like it could create significant friction.
It absolutely does. It can result in misaligned study protocols where different teams have different ideas about what they're actually measuring or validating. It leads to trial inefficiencies, validation deficits.
Wasted effort.
Potentially, yes. And it can even derail regulatory acceptance. There's been cases, for instance, where different agencies like the EMA in Europe and the FDA in the US interpret a seemingly simple term like monitoring quite differently in their guidance.
Wow.
This lack of a shared understanding creates friction, slows down progress. Imagine trying to build a complex machine when half your team calls the same crucial part by a different name. It just doesn't work well.
So what's the solution? We need like a Rosetta Stone for digital health terms.
Exactly. And thankfully, groups like EFPIA, the European Federation of Pharmaceutical Industries and Associations, and others have been working hard on this. They've proposed a harmonized framework with some key definitions to try and get everyone on the same page.
Okay, give us the highlights. What are the key terms we need to get straight?
All right. So first, digital health technology, DHT. That's pretty straightforward. It refers to the actual devices and software used for health data collection, the wearables, the app, the sensor.
The tools.
The tools, exactly. Then, digital measure. This is often used interchangeably with clinical outcome assessment, or COA, when derived digitally. It's a measure reflecting how a patient feels, functions, or survives, derived from DHT data.
Okay, the thing being measured reflecting patient state.
Right. Then, digital endpoint. This is more specific. It's a precisely defined variable derived from DHT data that's statistically analyzed to answer the main questions of the clinical trial, like, does the drug work?
So the measure used for the trial's conclusion.
Precisely defined for that purpose. And crucially, we need to distinguish these from ECOA, which stands for Electronic Clinical Outcome Assessment.
How's that different?
ECOA typically refers to taking a traditional clinical outcome assessment, maybe an old paper questionnaire or a simple diary, and just digitizing it onto a tablet or phone. It wasn't born digital. A purpose-built digital measure or endpoint is designed from the ground up to leverage the capabilities of the DHT. It's an important distinction.
Got it. And this leads us to another really critical distinction, maybe the most important one. Is the thing you're measuring with the DHT a biomarker or a clinical outcome assessment, COA? Because that a measure can kind of straddle both.
They absolutely can, and this is where it gets nuanced but incredibly important. Think of it as a spectrum. On one end, you have clear biomarkers. These are physiological measures, things like an ECG reading from a watch, blood glucose from a continuous monitor, or maybe temperature. They measure a biological process.
Okay, purely biological.
Right. On the other end, you have clear COAs. These are functional or symptom-based measures derived from DHTs. Think gait speed, calculated from a wearable, tremor intensity measured by a phone's sensors, maybe self-reported fatigue scores entered into an app. They reflect how a patient feels, functions, or, is able to function in their life.
How they experience the disease.
Exactly. What's fascinating is that some measures, like gait velocity, can actually be both, depending on the context and how you frame it in the trial.
How so? Why does this distinction matter so much? It sounds like more than just semantics.
Oh, it's far more than semantics. It's crucial because biomarkers and COAs demand very different types of evidence and different validation strategies to get regulatory approval. The bar is set differently.
Ah, okay. Different proof needed.
Exactly. For example, this measure SV95C in Duchenne muscular dystrophy, which we'll definitely talk more about.
Right.
It can be framed as a COA because it reflects functional ability, how well someone can walk at their peak. But you could also look at it through a biomarker lens as a physiological proxy for muscle function decline.
Interesting. Any other examples?
Sure. Think about activity counts, like step counts, measured by a wearable in patients with congestive heart failure. If you're using that activity level primarily to predict mortality risk, it's really acting more like a biomarker in that context.
Okay.
Whereas if you're measuring tremor or bradykinesia, slowness of movement in Parkinson's disease, using the smartphone sensor, whether actively during a task or passively in the background, your goal there is usually to capture the severity of those symptoms as experienced by the patient. That leans heavily towards being used as a COA.
So the classification directly dictates the validation you need to do.
Precisely. For biomarkers, the primary focus of validation is analytical validation. You need to rigorously demonstrate the reliability, accuracy, precision of the measurement itself. How well does the device actually measure the biological thing it claims to measure? You need to characterize the noise.
Technical performance.
Right. For COAs, you also need that analytical validation, but you additionally need robust clinical validation.
Which involves?
Which involves showing how changes in that digital measure actually align with how the patient is feeling or functioning overall with their well-being. This often requires linking the digital measure to other established clinical assessments or anchors. It involves patient interviews, qualitative research, and longitudinal evidence showing the measure changes meaningfully over time as the disease or treatment progresses.
Anchoring it to human relevance.
Perfectly put. without anchoring these incredibly sensitive digital measures to human relevance, essentially. Proving that a change detected by the device truly matters to a patient's life, they risk failing as COAs in the eyes of regulators, even if they're analytically perfect.
So the strategic takeaway here seems crystal clear. You absolutely have to decide early on whether you're positioning your digital measure primarily as a COA or a biomarker.
Yes, early and deliberately.
Use multi-stakeholder frameworks, involve patients early, and build the specific evidence package required for that classification. That precision is paramount for regulatory success, isn't it?
It underpins the entire development strategy. Getting that classification right guides everything else you do, from study design to the final analysis.
Okay, now this leads perfectly into another crucial concept, one that's essential for actually getting these digital endpoints accepted and used, defining what's meaningful.
Ah, yes, the meaningful change threshold.
Right. Even with all this incredibly precise, continuous data, regulatory acceptance, especially if you want to make claims on a drug label, hinges on defining what level of change actually matters to patients. And this seems way more complex for these continuous, data-dense digital endpoints compared to, say, traditional questionnaires.
It absolutely is more complex, but it's non-negotiable. It's about translating statistical significance into clinical relevance. It's not enough to show a change. You have to show a change that patients perceive as beneficial or important. That's the core idea of the meaningful change threshold, or MCT. The precise definition is the magnitude of change in an endpoint measure perceived as important to patients.
The patient's voice in the data, essentially.
Exactly. It grounds the endpoint in patient experience.
So how on earth do you figure that out? For something like continuous gait speed measured 24/7, it's not like asking, did you feel better, yes or no?
No, it's definitely more nuanced. It requires a thoughtful, multi-pronged approach, typically using three complementary methodologies.
Okay, what's the first one?
First, anchor-based approaches. This is where you relate changes in your digital measure, say SV95C, to changes in other established clinical measures that are already considered meaningful. These are your anchors.
Like what kind of anchors?
Could be clinician global impression scales, where a doctor rates the patient's overall change, like CGI improvement, or it could be changes in performance on traditional functional tests, like the 6-minute walk test, or even patient-reported outcome measures, like a quality of life questionnaire. You look for the amount of change, the digital measure, that corresponds to a change deemed meaningful on the anchor.
Okay, linking it to established benchmarks, what's the second approach?
That's distribution-based methods. These are more statistical. They use metrics like the standard deviation, SD, of the measure, or the standard error of measurement, SEM, which quantifies the measure's inherent noise or variability.
Right.
You can use these stats to calculate thresholds like the minimum detectable change, MDC, which tells you the smallest change that is likely real and not just random fluctuation or measurement error. It helps establish the floor for what could possibly be meaningful.
So separating the signal from the noise.
Precisely. And the third approach.
Third is expert consensus, often using something like a Delphi panel method.
How does that work?
You bring together a diverse group of clinicians who treat the disease, researchers, importantly patients or patient representatives, maybe caregivers too. You present them with data and scenarios, and through structured rounds of feedback and discussion, you work towards building a consensus on what magnitude of change in the digital measure they collectively consider to be clinically meaningful. Their combined experience and perspectives are invaluable. So the real power, then, is in combining all three methods, anchor-based, distribution-based, and expert consensus, to define that threshold. That's what you call triangulation, right?
Exactly. Triangulation is absolutely key. Relying on just one method can be misleading. When results from all three approaches point towards a similar range for the meaningful change threshold, you have much greater confidence in that value.
And you mentioned patient input. How central is that?
Increasingly central,
Yeah.
and rightly so. While clinician assessments like CGI are valuable anchors, there's a strong push towards using patient-reported impressions, asking the patients directly about the perceived change, like PGI, patient global impression, as the primary anchor whenever possible.
Because ultimately it's about what matters to them.
Exactly. They're the ones living with the condition day in, day out. Their perspective on what constitutes a meaningful improvement is paramount.
Now, what about the specific nuances of this continuous, high-frequency data? You hinted that traditional methods for analyzing change don't quite fit perfectly.
Yeah, they don't always translate directly. Many traditional methods for defining meaningful change were developed for COAs measured episodically, maybe once at baseline and once at the end of the study. With continuous data, you have this incredibly rich longitudinal trajectory for each person. So analysis often needs to be tailored. You might look at responder responses differently, perhaps defining response based on achieving a certain change sustained over time. Or you might use more sophisticated longitudinal modeling that focuses more on individual level change trajectories rather than just comparing group means at fixed time points.
So it's moving beyond simple averages.
Yes, it allows for a much richer, more dynamic picture of how individual patients are responding and changing over the course of a trial.
This brings us perfectly to what sounds like a landmark achievement, really tying all these threads together. The SV95C case study in Duchenne muscular dystrophy, DMD.
Yes, SV95C, a really pivotal example.
So first off, remind us, what exactly is SV95C and why is it so important for understanding how these concepts, validation, meaningful change play out in practice?
Okay, so SV95C stands for the stride velocity 95th percentile. It's measured using an ankle-worn wearable device called Actimeo, typically worn continuously. What it captures is the speed at which the fastest 5% of a person's strides occur during their daily life.
Not the average speed, but the peak speed.
Exactly. It's designed to capture peak ambulatory capacity, that burst speed, rather than just average walking speed. And this peak performance is thought to be a particularly sensitive indicator of functional ability and how it changes in boys and young men with DMD.
Okay. And how did this measure go from being, an interesting concept to actually getting regulatory qualification? That must have been an incredibly rigorous journey.
It was indeed a multi-year effort involving meticulous validation across several key domains. It really started with extensive analytical validation.
Proving the tech worked accurately.
Precisely. In controlled lab settings, the Actimeo device showed remarkable accuracy, about 98.7% accuracy in just detecting strides correctly. And when measuring stride speed, the mean error was tiny, only about 1 centimeter per second compared to a gold standard motion capture system.
Wow, that's very accurate.
It is. And importantly, they translated that to the field. In real-world use with DMD patients, they compared distances estimated from SV95C data over periods to the standard 6-minute walk test distance measured in the clinic. The error was incredibly small, around 0.24%, which is like being off by less than a meter over hundreds of meters walked.
That precision sounds remarkable. How reliable was it over time? Did it measure consistently?
Excellent reliability. They looked at month-by-month consistency using the intraclass correlation coefficient, or ICC. It was more than 970 overall, which is considered excellent.
Very high.
Extremely high. And importantly, even for the younger boys in the studies, aged 5 to 7 years, the ICC was still above 0.95. This showed it was reliable across different age groups within the target population.
And did they figure out how much wear time was needed?
Yes, that was key. They did analyses and found that you needed at least 50 hours of wear data per assessment period to keep the measurement variability below about 7%. They recommended aiming for 180 hours per period for optimal reliability.
And did the patients actually wear it that much?
Compliance was surprisingly high. In their studies, 100% of the participants met that minimum 50-hour threshold, and a very high 88% met the recommended 180-hour target.
Okay, so analytically, it's sound, accurate, reliable, feasible wear time. But what about the clinical validation? Does it actually measure something clinically relevant for DMD patients? Does it track the disease?
Absolutely. That was the next critical hurdle. They showed strong convergent validity,
Meaning?
meaning that changes in SV95C correlated well statistically with changes in traditional clinical outcome assessments used in DMD trials, things like the 6-minute walk distance, 6MWD, the North Star Ambulatory Assessment NSAA score, and the time taken to climb 4 stairs.
So when SV95C went up or down, those other measures tended to move in the same direction.
Exactly. Across multiple studies and time points, they showed these measures move together, suggesting SV95C was indeed capturing a related aspect of ambulatory function.
And could it tell the difference between, say, healthy kids and kids with DMD?
Yes, the known group's validity was very clear. They compared SV95C values between healthy control boys and boys with DMD. The difference was stark. The median SV95C in healthy controls was around 2.7 meters per second, while in DMD patients it was significantly lower, around 1.6 meters per second.
A clear distinction.
A very clear distinction, confirming its ability to differentiate between these clinical states, which is fundamental for any useful endpoint.
Okay, but maybe the most important part, how responsive was it? Could it detect changes due to treatment or just the natural progression of the disease, and how quickly?
This is where SV95C really shone. It proved to be extremely responsive. For instance, in patients starting on corticosteroids, a standard treatment, it detected improvements in average increase of about 0.09 meters per second in SV95C within just 3 months.
Wow, that fast.
Yeah. And conversely, in patients on stable steroid doses, it effectively tracked disease progression, showing a median decline of about 2.5% at 3 months, which increased to about 12.8% decline over 12 months.
But detecting change at 3 months, that sounds much faster than usual.
That was the crucial finding. SV95C was able to detect statistically significant change, both improvement and decline, as early as 3 months. This was a major advantage compared to traditional endpoints like the 6MWD, which often require 9 months, maybe even 12 months, to reliably show a change in DMD trials.
Detecting change that much earlier, that clearly offers a huge advantage. What are the implications of that speed?
It's genuinely game-changing for drug development in this space.
Yeah.
Detecting a treatment effect sooner means clinical trials can potentially be shorter, maybe require fewer patients, making them less expensive and faster to run. And for patients, obviously, it means potentially getting access to effective new treatments much faster. Plus, doctors might be able to adjust existing therapies more promptly based on objective data about how a patient is responding. It really accelerates the entire pipeline.
And did they establish what a meaningful change was for SV95C, that threshold we talk about?
Yes, they tackled the MCT. Using a combination of those anchor-based methods, linking SV95C changes to changes in NSOA scores and patient-clinician global impressions, and distribution-based methods, looking at sentiment and SD.
The triangulation approach.
Exactly, using triangulation. They proposed a threshold for a clinically meaningful decline in the range of -0.10 to -0.20 meters per second. This helps researchers and clinicians interpret whether a measured drop in SV95C is likely just noise or represents a change that actually matters to the patient's function.
How did the patients and the doctors react to using this wearable? Was there good uptake? Were people willing to wear it?
They were actually very receptive, which is crucial for implementation. Surveys conducted alongside the study showed that around 58% of DMD patients and their caregivers considered top walking speed what SV95C measures as a highly prioritized outcome.
Okay, so it measures something they care about.
Yes, and importantly, about 70% said they preferred wearable-based real-world monitoring over frequent clinic visits for tests. A remarkable 75% said they were eager to wear the device in a trial, with almost half willing to wear it continuously.
That's strong acceptance.
Very strong. And healthcare professionals involved in the trials also reported positive attitudes, recognizing the value of objective, continuous data and finding the device feasible to use in practice.
Okay, so you've got analytical validation, clinical validation, responsiveness, meaningful change defined, patient acceptance. Which brings us to the big reveal. The regulatory milestone.
Yes, the culmination of all that hard work. In July 2023, the European Medicines Agency, the EMA, formally qualified SV95C.
Qualified it as what?
They issued a positive qualification opinion for SV95C as a primary efficacy endpoint for clinical trials in Duchenne muscular dystrophy for ambulatory children aged 4 years and older. This was the first time ever that a digitally derived measure received such qualification as a primary endpoint from a major regulatory agency like the EMA.
The first ever digital primary endpoint. That is huge.
It's truly a groundbreaking achievement, a massive milestone, not just for DMD research, but for the entire field of digital health and clinical trials.
Why is this single qualification so significant? It's obviously great for DMD, but the impact feels broader.
Oh, it's much bigger than just DMD. SV95C's success sets an incredibly powerful precedent. It demonstrates definitively that novel digital endpoints derived from wearables worn in the real world can meet the extremely high bar of rigorous regulatory scrutiny needed for primary endpoint status.
It proves it can be done.
It proves it can be done. It provides a tangible blueprint, a roadmap for how other sponsors can develop and validate their own digital endpoints across countless other diseases. It shows that the path from raw, sometimes messy, real-world data all the way to a qualified, impactful clinical endpoint is now clearer and, importantly, validated by regulators.
Oh. Okay, this deep dive has really shown us the incredible potential, but also the complexities of using DHTs in clinical trials. To kind of recap the essentials for success, It seems to boil down to having really robust strategies for handling that inevitable missing data.
Absolutely crucial.
Thoughtful BYOD design, balancing convenience with rigor,
Mm-hmm.
using clear, harmonized terminology so everyone's speaking the same language.
Mm-hmm.
Having that precise understanding up front of whether your measure is fundamentally a biomarker or a COA,
Yeah.
because that dictates the evidence you need.
Can't stress that enough.
And maybe most critically, defining those scientifically derived,
Yeah.
but ultimately patient-centered, meaningful change thresholds.
You've nailed the key ingredients. And the SV95C story, it really is a beacon. It perfectly illustrates that whole journey from raw, high-frequency sensor data to a fully qualified, impactful clinical endpoint. It proves that digital health can genuinely accelerate drug development across a vast range of health conditions. We're really seeing a fundamental shift in how we approach clinical evidence generation.
It really makes you wonder, doesn't it? As these digital health technologies become even more seamlessly integrated into our everyday lives, how might this whole paradigm shift in clinical trials fundamentally change the speed at which new treatments actually reach patients? And maybe even how it reshapes our own understanding of what health truly looks like, beyond just snapshots from clinic visits.
A profound thought, capturing life as it's lived.
Definitely something for all of us to mull over. Thank you so much for unpacking all of that today.
My pleasure. It's an exciting time.
Join us next time for another